These cells can form the tissues, which in case humans, a group of cells can form four different type of tissues called as epithelial tissues, connective tissues, muscular tissues, and nervous tissue. And these tissues in turn forms the organs, and organs form the organ systems, and organ system eventually form one complete organism or one complete individual, right? So the organism is formed of a small basic unit which is called a cell. So it is about the cell. Actually, biology is full of exceptions, right? So here also there is one exception which is called as viruses. Viruses do not have actually the cellular structure. Actually, virus structure composed of an RNA which is covered by a protein coat. So that we do not consider viruses as truly living. In fact, we consider them as connecting link between the living and non-living. Because viruses, when infect the human beings or any host, they will attain the living state. That means they can multiply in the living organism or that host. But when they live in the environment, they behave as non-living things. So that we consider these viruses as connecting link between the living and non-living things. Okay. Now, depending upon the number of cells present in an organism, we classified the organisms into unicellular organisms and multicellular organisms. Unicellular organisms, as the name suggests, these organisms has the only single cell. For example, the examples of unicellular organisms are bacteria and protozoans, whereas the multicellular organisms has many cells. The examples for multicellular organisms include fungi, plants, and animals, starting from porifera to chordates in the animal kingdom. Friends, this is a very important point. Unicellular organisms, although they have only single cells, they can exist independently without depending on any other organisms or any other cell. And also, apart from that, they can perform the essential functions of life. For example, digestion, respiration, circulation. All those functions can be performed by only that single cell only. So that we can consider that cell is considered as a structural and functional unit of life. Anything less than a complete structure of cell does not ensure independent living. For example, viruses do not have the complete structure of cell, so we didn't consider them as a complete living organism, right? So, friends, now let's understand how the cell have been discovered, how the cell came into limelight. Uh, in the year 1665, Robert Hooke, named a scientist, observed different types of cork cell under the microscope. Actually, he observed honeycomb-like structures under the microscope, and he named those structures as cell. And he mentioned all those details in his book called as Micrographia. But here, think we need to remember one thing, which is called as he actually observed the dead cells, because cork cells are dead. So the credit of dead cell discovery goes to Robert Hooke, and in the year 1675, a scientist known as Anton von Leeuwenhoek, actually he observed RBC, protozoan, and different types of samples under the microscope. So he observed the first live cell. So Anton von Leeuwenhoek got the first credit of discovering the live cell, whereas the Robert Hooke got the credit of discovering the dead cells. Later, after some years, in the 1831, Robert Brown, named a scientist, discovered the nucleus, which is present inside the cell at the central location. So, although there are many scientists in the cell biology, these three scientists have been played a major role because they have identified the cell and nucleus. So, these three scientists are most important. Friends, now let's understand about cell theory. What exactly mean by the cell theory? Actually, cell theory to propose cell theory, there came the two scientists called as Matthew Sweden and Theodor Schwann. In the year 1838, is a German botanist. His name was Matthew Sweden. Actually, he was a botanist, so he observed different kinds of plant tissues. He observed and studied different type of plant tissues and stated that all the type of plant tissues composed of plant cells. This was a statement given by Matthew Sweden. And at the same time, within a year, that is 1839, a British zoologist, his name is Theodore Schwann. Actually, he was a zoologist. He studied different types of animal cells. And he stated that all the animal cells had a thin outer layer, which is today known as the plasma membrane or cell membrane. Remember here, he told that animal cells contain plasma membrane around the cell. And also, fascinating thing that being a zoologist, he also studied the plant cells also. We need to remember this point because in a lot of examinations, it will be framed as a question also. Being a zoologist, Theodor Schwann also studied plant cells. And he stated that plant cell is contains cell wall, which is a unique characteristic of unique character of plant cells. Okay, remember this point. On the basis of this, Schwann proposed the hypothesis that animals and plants bodies are composed of cells and product of cells. Products of cells mean the products which are released by cells, for example, proteins or ACM or any other products which are being released by those cells or with those plant cells or animal cells. Okay, but the problem is actually what happened means that these Schleiden and Schwann are belong to different places, but actually they stated the same thing, but they were unable to find out how the new cells were arising. They were used to believe the theory of abiogenesis, that is, all living organisms arise from non-living things, somewhat like that. So at that time, a scientist came. His name is Rudolf Virchow. He gave in a statement which is known as omnicellular e-cellular. It is a phrase. Actually, it means all the living cells arise from pre-existing cells. So this statement had what we say uh, uh, played the major impact in this cell theory. So after Rudolf Virchow combined this statement in this cell theory and given the cell theory a final shape. So cell theory as understood today is all living organisms are composed of cells and products of cells. This point will be given by Schwann and Theodor Schwann and Matthias Frieden. And also he included his point also, all cells arise from pre-existing cells. Okay. Friends, it was a structure of onion peel cells under the microscope. Actually, it was the image from light microscope, not from electron microscope. So the structural details are not such clear, but we can able to what we say observe the cell wall of this plant cell. See these hexagonal structures. These are called as cell wall of plant cells. Inside the cell wall, we can see the cell membrane, and inside the cell membrane there is cytoplasm. At the center we have the nucleus, and also different types of cell organelles also present in the cell cytoplasm, right? So we will discuss when we when we are studying about the plant cells, all those things. Okay. Now let's look at the overview of the animal cell. Actually, I don't have the animal cell picture, so I draw it simply. So actually, the outer membrane of animal cell is cell membrane. Animal cells do not have any cell wall. And also, as we all know, animal cells contain cytoplasm. In the cytoplasm, they have different types of cell organelles like endoplasmic reticulum, Golgi, mitochondria, and all those things. Lysosome, such that organelles will be present in the cytoplasm, and also nucleus will be present in the center. In this nucleus, we have DNA. So it's about the overview of animal cell, but we will study in depth when we studying the animal cell. Okay, friends. Now let's try to understand the classification of cells depending upon the structure of nucleus. Initially, we have understand unicellular and multicellular organisms. And that it is based on the number of cells. Unicellular organisms contain single cell, whereas multicellular organisms contain multiple cells, right? But here we are classifying the cells depending upon the nuclear structure. As we all know, nucleus is present in the center of the cell. This nucleus in turn contains DNA. Okay. So what happens in the prokaryotes means the first classification is prokaryotes and the second classification is eukaryotes actually. In prokaryotes, as the name suggests, pro means primitive and karyotes means nucleus. These organisms contains a primitive type of nucleus. That means its nucleus is not bound by any what we say nuclear membrane or nuclear envelope. So such type of organisms are called as prokaryotic organisms. The best example of prokaryotic organisms is bacteria. Although there are many differences, we will discuss. The major difference is depending upon the nuclear membrane. When it comes to eukaryotes, eu means true and karyotes means nucleus. Actually, these are the advanced type of organisms when compared to prokaryotes. In this type of organisms, nucleus is surrounded by well-defined nuclear membrane. Okay, here there is no well-defined nuclear membrane, but in case of eukaryotes, there is well-defined nuclear membrane. The best example for eukaryotic organisms are protozoans, fungus, plants, and animals. So it is a primary difference between the prokaryotes and eukaryotes. Although there are many differences, we will see what are those differences between prokaryotes and eukaryotes. Friends, now let's understand some more differences between the prokaryotes and eukaryotes. We have seen the first difference. Prokaryotes do not have well-defined nuclear membrane. Eukaryotes have well-defined nuclear membrane, right? The second difference is in the prokaryotes, the nuclear membrane, that what we said, the genetic material, the DNA, do not have any histone proteins. It is randomly arranged at the center, which is called as genophores. Whereas in the eukaryotes, the DNA, that is the nuclear membrane or the genetic material, is associated with a type of proteins called as histone proteins. So they form the compact structures called as chromosomes in the case of eukaryotes. But in the prokaryotes, there is no such structures called as chromosomes. The DNA is just randomly placed in the center, which is called as genophore or nucleus. In the prokaryotes, the DNA content is very less, and the eukaryotes, the DNA content is high. And also in the prokaryotes, the major difference is they do not have any membrane-bound cell organelles such as Golgi, any. They do not have any such organelles. They only have a organelles called as ribosomes. And such ribosomes are also type 70th type of ribosomes. We will see what exactly mean by the type of 70th type of ribosomes in few minutes. Actually, remember this point first. Uh, Actually, try to remember this point. Prokaryotes have only 70th type of ribosomes. Apart from this, they do not have any membrane-bound cell organelles. Okay, and their ribosomes also they have 70th type of ribosomes. Whereas in the case of eukaryotes, they have membrane-bound cell organelles also, and also they have ribosomes. That too, 80th type of ribosomes. So it is these are the major differences between the prokaryotes and eukaryotes. And we have seen the examples. Prokaryotes include bacteria, whereas eukaryotes include protozoans, fungus, plants, and animals, right? So friends, it's the basic overview. Prokaryotes contain only the ribosomes, the 270th type of ribosomes present in the cytoplasm, whereas eukaryotes contain ribosomes, the 280th type of ribosomes. These ribosomes can be present in the cytoplasm, rough endoplasmic also in the rough endoplasmic reticulum, that is RER also these 80th type of ribosomes are present, and in the mitochondria also these 80th type of ribosomes are present in the case of eukaryotes. Okay, and also the organelles we have seen that just now we have seen eukaryotes have membrane bound organelles, right? So now let's see what are those organelles. Those are endoplasmic reticulum, we call as ER, Golgi complex, we simply say Golgi, lysosomes, mitochondria, microbodies. Microbodies are actually the fragments of endoplasmic reticulum only. Vacuoles, remember these vacuoles are present only in the case of plant cells. And chloroplasts. Chloroplasts are the type of plastids. These are also present only in the case of plant cells only. And the centrosome. Centrosome is a round structure. In the centrosome, there are two cylindrical structures called as centrioles. And remember this point, centrosome or centriole is present only in the case of animal cells only. In place of centrosome and centrioles, in plant cells, they have called a structure called as MTOC, which is called as microtubule organizing complex. Actually, it plays a key role in the formation of spindle fibers during the cell division. So we will discuss all these points. Just I am giving the overview about organelles present in the case of eukaryotes. Okay. Now first let's study about the prokaryotic cell, how its structure would be, and what are the shapes and the example, and all those things we will study about the prokaryotic cell. So, as I have told, cells actually differ greatly in their size, their shape, and activities. Here, here they are comparing about the sizes of different prokaryotes and eukaryotes. For example, mycoplasma is a type of prokaryote. It is about zero point 0.3 micrometers in length. See how small it is. Whereas, if you observe the size of bacteria, it is 3 to 5 micrometers in length. 3 to 5 micrometers actually. If you observe the size of multicellular organisms, for example, human RBC, it is about 7 micrometers in diameter. Whereas in the egg of an ostrich, it is actually the largest isolated single cell. Remember this point. Egg of an ostrich is considered the largest isolated single cell. If you consider the longest cell, nerve cells can be considered as the longest cells in our body. Okay. Actually, here what they are trying to explain is cells, even though all are the basic unit of life, they exhibit in different sizes. So here we see mycoplasma is zero point 0.3, bacteria is 3 to 5, RBC is 7 micrometers, and the egg of an ostrich itself a largest cell, and also nerve cell is longest cell. So they're explaining cells exhibit in different sizes. And in the next slide, they are explaining about cells also, explaining about different shapes of cells. They may be disc-like, polygonal, columnar, cuboidal, thread-like, or even irregular in some cases. Actually, the shape of cell is associated with the function they perform. Actually, for example, RBC are round. RBC shape is round and biconcave. They accommodate this shape because they need to accommodate the hemoglobin and oxygen during the transport of gases. So they exhibit in the round and biconcave shape. Whereas in the WBC, they are amoeboid in shape because they will show a movement known as amoeboid movement or phagocytosis during the immune responses. And in the columnar epithelial cells, they are long and narrow because they need to absorb the nutrients from the intestine. And the nerve cells are very long because they need to transmit the action potentials during the nerve impulse conduction. And when we see tracheids actually a part of xylem. Tracheids, vessels, they play a major role in the transport of water actually. Mesophyll cells are present in the leaf. Actually, they are tightly packed. They're actually round and oval also. They contain chloroplast also. Okay. So here they are explaining about the shapes of different types of cells. Now first let's study about the prokaryotes. Then we will move on to the eukaryotes. Just now I have explained the prokaryotic cell is the bacteria is the best example for prokaryotic cells, right? Apart from bacteria, we can consider blue-green algae also, which is called as cyanobacteria. Mycoplasma, just now we have seen zero point 0.3 micrometers in length, the smallest cell, and PPLO, which is called as pleuropneumonia-like organisms. So all these organisms comes under the category of prokaryotic cell. Prokaryotic cell means the major difference is this cell nucleus is not bound by any membrane not bound by any nuclear membrane, right? That is the primary difference in the prokaryotic cell. These prokaryotes are generally very smaller and multiply more rapidly than the eukaryotic cell. Actually, they will multiply more rapidly. For example, if you take the example of bacteria, their generation time is approximately 20 minutes only. Within 20 minutes, one bacteria can form two. They will divide, and in the 40 minutes, these two can form the four. So it is the generation type of bacteria. So that they have mentioned they will multiply more rapidly than the eukaryotic cell. For example, if you take the human cell, it takes around 24 hours to divide. If you take the yeast cell, it takes around 90 minutes to divide. So when compared with eukaryotic cell, the bacteria cell generation time is very small, very short, right? So this is the point. Actually, these bacteria or these prokaryotes also exhibit in different types of shapes also, and also shapes also. There are majorly four different types of shape. For example, bacillus. Bacillus means if the bacteria exhibits in rod-like shape, then we call such type of bacteria as a bacillus bacteria. For example, we see lactobacillus bacteria, which use it to milk, which can be used to convert milk into curd. And if the bacteria is spherical in shape, then we call such bacteria as coccus bacteria. For example, streptococcus, staphylococcus, such type of bacteria is called as coccus bacteria. Okay. Next, if the bacteria exists in comma shape, then we call it as vibrio bacteria. For example, the bacteria which causes cholera, that is vibrio cholera, right? It actually exhibit in comma shape. The fourth category is spiral shape. And for example, actinomycetes, such group. If the bacteria fits in spiral shape, we call such bacteria as spirillum. Okay. So these are the four different basic shapes of bacteria. Friends, now let's understand the organization of prokaryotic cell, how the prokaryotic cell is organized. Actually, organization of prokaryotic cell is fundamentally similar, even though they exhibit in wide variety of shapes and functions. Actually, all the prokaryotic cell wall is made up of a chemical known as peptidoglycan. Peptidoglycan is actually formed of NAG and NAM. NAG means N-acetyl glucosamine. NAM is N-acetyl muramic acid. Actually, these two are glycosaminoglycans. Remember this thing, peptidoglycan is formed of N-acetyl glucosamine and N-acetyl muramic acid. Okay, this cell wall is surrounded inside to the cell wall is the presence of cell membrane. Except in the mycoplasma, there is cell membrane present in all the case of all prokaryotes. Okay, next fluid filling matrix is called as cytoplasm, as we all know, and there is no well-defined nucleus. And the genetic material is basically naked. That means there is no nuclear membrane in the case of prokaryotes, and there is no organelles present in the case of prokaryotes like the ones present in the eukaryotes. It only have ribosomes, that to 70th type of ribosomes. And in the presence of and in the prokaryotes, there is a the reserved food material is stored in the form of inclusion bodies. Okay, and also in prokaryotes, the plasma membrane is folded inside to form a structure called as mesosomes. Mesosomes helpful in the different types of functions we will see further. Actually, remember this point, the plasmid folded inside to form structures called as mesosomes, just like the crystal of mitochondria. And also, this is the important point we need to remember. Plasmid is actually extra chromosomal material present in the bacteria. Actually, this plasmid present in the cytoplasm, it will provide the resistance for various antibiotics. If the bacteria has this plasmid, it may provide the resistance for different types of antibiotics which can be used to kill bacteria. And also, this plasmid plays a very crucial role in RDNA technology. We insert our gene of interest into this plasmid and we will allow this amplification in this bacteria so that we get our desired protein or desired product in this RDNA technology. So that's what they have explained about plasmid. It can provide unique phenotypic characters. One such character is resistance to bacteria, and it plays a very important role in RDNA technology. You will be studying all those details in biotechnology in RDNA technology. Okay. And also now let's understand the cell envelope of these prokaryotes. Actually, as we till now we have understand. Actually, bacteria cell is surrounded by cell membrane, which is called as plasma membrane. Outside the cell membrane or plasma membrane, we have cell wall. Bacteria has cell wall. This cell wall is made up of peptidoglycan, which is formed of N-acetyl glucosamine and N-acetyl muramic acid. And outer to this cell wall, there is another layer which is called as glycocalyx. Glycocalyx is a glycoproteinaceous layer. Actually, it forms the covering for the bacteria so that it can provide extra resistance from the host enzymes or host cells or macrophages when it infect the host cell. Okay. Although each layer, glycocalyx, cell wall, and plasmid, each have a different function, they together form a single protective unit. They together act as a single protective unit. Okay. Actually, this glycocalyx in some cases exist as a loose sheet. Then we call it a slime layer. In some cases, it can exist as a thick and tough sheet. Then we call it as a capsule. Okay. Remember this point. It is very important. Glycocalyx, if it is loose, then we call it as a slime layer. If it is thick and tough, then we call it as a capsule. Okay. Cell wall, as we all know, it provides the shape for the cell and provides a strong structural support and prevent bacteria from bursting or collapsing. Okay. In case of bacteria, cell wall is made up of peptidoglycan. I'm repeating it every time because you can remember it easily. Next, as we all know, plasma membrane, it is selectively permeable in nature. So actually, plasma membrane is similar to eukaryotes only. Whatever the type of organism, prokaryote or eukaryote, the type of plasma membrane is same, which is phospholipid bilayer. We will see those while studying the plasma membrane structure. Just now I have told about mesosomes. These are the structures formed by the extension of plasma membrane inside the cell. Okay. Actually, these mesosomes are in the different forms: vesicles, tubules, and lamellae. They will help in the formation of cell wall. They will help in the DNA replication and its distribution to daughter cells. They also help in the respiration. They have different enzymes on those and also different secretion process and to increase the surface area of plasma membrane, and also it has different types of enzymes on it also. Okay. And also, for example, cyanobacteria, which is called as blue-green algae, they have pigments in the structures, in this membrane extension. What we say, these structures. So that we call such structures as chromatophores also. Okay. Uh, these are actually analogous to mesosomes only, in which but they contain pigments and they perform photosynthesis also. So that we can call those as chromatophores. When it comes to motility of prokaryotic cell, actually for motility we know there is a presence of flagella, but here there is no cilia. In bacteria there is presence of flagella, but the type of flagella present in the bacteria is very much different when compared to the flagella which is present in eukaryotic cells. Okay, and this flagella structure is composed of three parts: filament, hook, and basal body. Filament is the longest portion, and the hook is the neck portion, and basal body is the basal part of the flagella. Okay, this what I was written here. Next, there are other structures also on the surface of the plasma membrane of bacteria, but they do not play a major role in motility. The first one is pilus. Pili, we also called as pili. These are elongated tubular structure. Actually, when compared to fimbriae, pili are somewhat long. Phyla or elongated tubular structure made of special protein called as phylin. So actually they play a major role in conjugation. Conjugation happens between two different types of bacteria to exchange their genetic material. Okay, conjugation, transformation, and transduction are the three methods to exchange genetic material in the case of bacteria. Fimbria are the small bristle-like structures present on the cell membrane of bacteria. Actually, they will help to attach bacteria to rocks which in the streams and also to the host tissues also. Okay, just now I have told phyla and fimbria are the surface structures only of the bacteria. They do not have a role in motility. And ribosomes, just now I have told, bacteria contain only this. The prokaryotes contain only the cell organelles called as ribosomes. These ribosomes also, that too 70th type of ribosomes they have. 70th ribosomes actually we need to understand one thing. Every ribosome is composed of two subunits, one large subunit and one small subunit. Here in the case of 70th type of ribosomes, large subunit is called as 50th type of subunit. And the small subunit is 30th type of subunit. Okay, remember this point. Every ribosome is composed of two subunits, one large subunit and one small subunit. Sorry, one large subunit and one small subunit. Here in the case of 70th type of ribosomes, large subunit is 50th and small subunit is 30th. Okay. And also we need to remember one point also here. During the process of transcription, that means production of mRNA from DNA. Actually, the mRNA comes into the cytoplasm. Onto this mRNA, many different type of ribosomes attached. So such structure we call as polyribosome or polysome. We observe this structure only in the case of prokaryotes, not in the case of eukaryotes. So this structure is called as polyribosome or polysome. The ribosomes of polysome translate this mRNA into proteins. Okay, actually, we need to remember one point. Actually, ribosomes are considered as a protein factory. Ribosomes helpful in the synthesis of proteins. They play a major role in the process of translation. Translation means formation of proteins. That means synthesis of protein is called as translation. Ribosomes play a major key role in the process of translation. Okay. Just now I have explained inclusion bodies. Inclusion bodies means prokaryotes store their food material in the form of inclusion bodies, and they do not have any membrane bound. They do not have any membrane. They lie free in the cytoplasm. Examples of inclusion bodies include phosphate granules, cyanofacial granules, and glycogen granules. And also certain green and pulpal photosynthetic bacteria has certain structures called as gas vacuoles. They will help in the process of buoyancy or floating. Okay, so it's all about the prokaryotic structure. Now there is a famous experiment called as gram staining. It is a technique actually used to determine or to differentiate a gram-positive bacteria and gram-negative bacteria. First, let's understand what exactly mean by the gram-positive bacteria and gram-negative bacteria. Till now we have discussed every bacteria is made up of a cell wall, right? That took peptidoglycan. Actually, depending upon the difference in the peptidoglycan layer, bacteria is divided into two types: gram-positive and gram-negative. In the case of gram-positive bacteria, it has very thick cell wall. Okay, gram-positive bacteria has very thick cell wall, and the gram-negative bacteria has very thin cell wall. Up to now, remember this point: gram-positive means has thick cell wall, gram-negative means has thin cell wall. Now, depending upon this difference, we uh, classify bacteria. We perform a test known as gram staining. Now, let's understand the procedure of or what we say protocol of this gram staining technique. First, we need to take a slide and we add the bacterial culture and we'll fix it. And then we will add a chemical known as crystal valate and we add iodine. Iodine is called as mordant because iodine help in retaining the structure to the bacterial cell wall. So when we add iodine, crystal valate and iodine form a complex. They will form a strong complex, crystal valate and iodine complex. Then after 1 minute or half minute, then we will wash with decolorizing agent, that is ethanol or acetone. At this time, what happens means gram-positive bacteria retain the color actually because their cell wall is very thin, very thick, right? Whereas in the case of gram-negative bacteria, their cell wall is very thin. So they, as soon as they wash with decolorizing agent, that is ethanol or acetone, the gram-negative bacteria start to lose this association, crystal valate and iodine. Okay, whereas the gram-positive bacteria, Gram-positive bacteria retain this color, whereas the gram-negative bacteria lose this color. Okay, so to stain this gram-negative bacteria, we stain with safranin again, and we will observe it under the microscope. So the result will be the gram-positive bacteria will be appeared in the purple color. The gram-negative bacteria will be appeared in pink color. In gram-positive bacteria will be retained the crystal violet and iodine. Gram-negative bacteria will be retained will lost that crystal violet and will be stained by safranin. So gram-positive bacteria will be appeared in purple color. Gram-negative bacteria will be appeared in pink color. Okay, so it is the reason. I have explained, have wrote the reason here. Gram-positive bacteria has a thick cell wall, hence it can retain the crystal violet. Gram-negative bacteria has a thin cell wall, hence most of the crystal violet is washed away while washing it with decolorizing agent like ethanol or acetone. So on the basis of this, we can classify the bacteria into gram-positive and gram-negative. Okay, and also one important thing is gram-negative bacteria also have a lipopolysaccharide layer, that is LPS layer on this cell wall. So it can provide the extra resistance for the gram-negative bacteria when it infects the host cells. So that's the reason why most of the pathogens infecting the humans are gram-negative only. They have LPS layer, so our host can't able to find the gram-negative bacteria such easily. Okay, whereas the gram-positive bacteria do not have any LPS layer. So friends, it's all about the prokaryotes, that is bacterial cell. It is the overview of the bacterial cell. So it is a cell membrane, that is plasma membrane. Outside of the plasma membrane, there is cell wall, right? Cell membrane is extended into foldings called as mesosome, and outside of the cell wall, we have a glycocalyx layer also. Uh, and also the only organelle present in the prokaryote is 70S ribosomes here, and the surface structure is pilus, it is very long, and this yellow color structure are fimbria help attached to any rocks, bristle-like structures, and it is a flagellum which is different from eukaryotes. It's all about, and it is a uh, chromosome or sorry, it is a genetic material which is randomly arranged in the center, which is called as nucleoid, and extra chromosomal material which is called as plasmid. Okay, it's all about the structure of bacterial cell or the structure of prokaryote. Okay, now let's move on. Friends, now let's study about the eukaryotic cells. Eukaryotic cells, as we have discussed earlier, it include protists, plants, animals, and fungi. Eukaryotes have extensive compartmentalization, that means the presence of membrane-bound organelles in the cytoplasm. Eukaryotic cells possess an organized nucleus, that is, the nucleus is bound by a nuclear membrane. Okay. In addition, eukaryotic cells have very complex locomotory and cytoskeleton structures. That means here they are talking about cytoskeleton, which is composed of microtubules, microfilaments, and intermediate filaments. We will discuss when we are discussing about cytoskeleton. Okay. Here the genetic material is organized into chromosomes. That means the DNA is coiled around proteins called as histone proteins and form a compact structures called as chromosomes. And also all eukaryotes have this structural, what we say, they are not identical actually. They say they have the same pattern, that is cell membrane, RER, Golgi, but they are not exactly identical. They are somewhat different. For example, in the case of plant cell, they have the cell wall, but in the case of animal cell, they do not have any cell wall. And also in the case of plants, they have large central vacuole and the plastids, but they are absent in the case of animal cells. In turn, animal cells contain centrosome and centriole. These centrosome and centriole are absent in the case of plant cell. Plant cell contain empty OC, that is microtubule organizing complex. Okay, these are somewhat differences between the prokaryotes and eukaryotes. Here they compared about the plant cell and animal cell. So it is the structure of plant cell and animal cell. If you observe the plant cell, it is a nucleus. Inside the nucleus, this is the nucleolus structure. Uh, what we say? Uh, outer the out From the outer membrane of nucleus, there is a structure called as RER, rough endoplasmic reticulum. We call. After the rough endoplasmic reticulum, there is a structure called as smooth endoplasmic reticulum, and also there is a Golgi complex. Golgi complex helpful in the glycosylation or modifications of proteins or any carbohydrates. And also there are certain membrane bound organelles also, mitochondria, chloroplast, peroxisome, and different types of cell organelles. For it is a large structure called as vacuole, and also Plasmodesmata is a structure which is helpful in the connecting of two plant cells. Lysosome contain very what we say acidic enzyme, hydrolytic enzymes, which can degrade all the cell. So it is the basic organization of plant cell. We will study this organization also. It is a basic overview of how the plant cell will be look like. Next, when compared to animal cell, when comes to animal cell organization is also almost same, but there are some minute differences. Animal cell do not have any cell wall, and animal cell have microvilli also. For in some cells, there is a microvilli for movement, and also animal cell have centrioles in the centrosome, and also animal cells do not have any plastids like chloroplasts, all those structures, and also what we say animal cells have different types of cell organelles like mitochondria, lysosome, uh, what we say Golgi, and all the cell organelles which are similar to that of plant cells. So there are one, two, three, or two to three differences between the plant cell and animal cells. Apart from this, almost the structure of the plant and animal cell is very much similar. Okay, friends. Now we are going to study about all the type of organelles which are present in the case of eukaryotic cell. First, let's study about the plasma membrane, which is present in all the prokaryotes and eukaryotes also, which is acting as the boundary for the cell. Actually, the detailed structure of membrane was not studied till the electron microscopy discovery, but our scientists have discovered what is the composition of our plasma membrane by studying the RBC. These studies show that cell membrane is mainly composed of lipids, that means phospholipids, and proteins. The major type of lipids that are present in the case of plasma membrane are phospholipid. Actually, they are arranged in bilayer, that is outer leaflet and inner leaflet, outer leaflet and inner leaflet. All the lipids are arranged within the, that means polar head towards the outside, that means their head groups towards the outside and the tail groups towards the inner part. We will see in the next slide in the structure of plasma membrane. This ensures that a non-polar tail of saturated hydrocarbons, this saturated hydrocarbon non-polar domain is protected from the aqueous environment actually. In addition to phospholipid, membrane also contains cholesterol. It plays a major role in the process of fluidity. That means our plasma membrane is not static; it keeps on moving. Biochemical investigations reveal that plasma membrane also contain proteins also and carbohydrates also. Proteins are but that what we say the composition of protein and lipid is different in different types of cells. For example, if you observe the RBC of human, there is 52% of proteins and 40% of lipid. It will be varied if you observe the different types of cells. But the thing is, every plasma membrane contain proteins, lipids, carbohydrates also, okay, and cholesterol also. And also, these proteins, depending upon the ease of extraction, they can be classified into two types: integral proteins and peripheral proteins. Integral proteins means these proteins can be integrated on the membrane, so they are hard to what we say extract. Whereas peripheral proteins are located on the surface or on the outer structure of the plasma membrane, so they are very easy to extract from the plasma membrane. Okay, so it is the basic structure of plasma membrane. Actually, it was the fluid mosaic model which was proposed by Singer and Nicholson in the year 1972. It is the widely accepted model for the plasma membrane. So according to this, the quasi-fluid nature, that is semi-fluid nature of lipid, enables lateral movement of proteins. See here, it is the integral membrane proteins, and it is the peripheral proteins. Peripheral proteins are located on the peripheral side. Integral proteins are embedded in the plasma membrane, right? The ability to move within the membrane, that is just now I have told, no, these proteins and cholesterol are not static, they can be moved. So this property is called as fluidity. Our plasma membrane is fluid, right? The fluid nature of membrane is important for many functions like cell growth, formation of intercellular junctions, secretion, endocytosis, that means taking the food material inside the cell or any components inside the cell, and cell division. So this fluid nature of plasma membrane plays a major role in very different type of functions, right? So actually, the major function of plasma membrane is to transport the molecules across it. So to understand transport, there are different types of transport. First one is passive transport. The second type of transport is called as active transport. Passive transport means, remember one thing, passive means Here we do not have here we do not use any type of energy. The molecules can move from their higher concentration to lower concentration. Okay. There are two different types of passive transports actually. The first one is simple diffusion and second one is facilitated diffusion. In the simple diffusion, actually the neutral molecules can move from their higher concentration to lower concentration through the plasma membrane. Here they are moving from uh, they are moving along the concentration from, that means from higher concentration to lower concentration. So that's why we are not using any energy or any ATP here, right? Whereas in the case of facilitated diffusion, here also the moment is from higher concentration to lower concentration only. But here the molecules which are moving are polar. As they are polar, the plasma membrane is non-polar. The plasma membrane cannot, what we say, allow these polar molecules to move through that. So at that point, there is a protein called as carrier protein. It will be helpful in the movement of these polar molecules from their higher concentration to lower concentration. Okay. So osmosis actually is also the same term, same process, but the term osmosis can be used to liquids like water or any solvent molecules. Active transport, when it comes to active transport, actually it is ATP dependent or energy dependent. Here, the movement of molecules is against a concentration gradient, that is from lower concentration to higher concentration. That means we need to use energy here. For example, uh, if we need to throw a stone to upwards, we need to put give some energy, right? Whereas if we throw stone from up to down, then we need no need to give any energy, right? So the same way in the active transport, we need to give energy. So such type of transport is called as active transport. The best example for this active transport is sodium potassium pump. It is present on the surface of cell. It can transport 3 sodium outside the cell and 2 potassium inside the cell. Okay. I hope you're clear about this passive transport and active transport. Friends, now let's discuss about the cell wall. As you all know, cell wall is a non-living rigid structure. It forms the outer covering for the plasma membrane of fungi and plants. In fungi, the cell wall is made up of chitin, whereas in the case of plants, the plasma membrane is majorly composed of cellulose. These are of eukaryotes, right? Fungi and plants, whereas in the case of prokaryotes, the bacteria cell wall is made up of peptidoglycan. Whereas the animal cells do not have any cell wall, right? A cell wall not only gives shapes, it can protect the cell from mechanical damage and it can protect from infection also. Cell wall also helps in cell-to-cell interaction, and it can provide the barrier for undesirable molecules from entering into the cell. It actually acts as a barrier also. So next point, they are talking about the composition of cell wall. Algae have a cell wall made up of cellulose, galactans, mannans, and minerals like calcium carbonate. Next, when it comes to plant cell, it composed of cell wall made up of cellulose, hemicellulose, pectin, and different types of proteins. Okay, it is a composition of plant cell wall and algal cell wall. Actually, the next point they are talking about the primary cell wall and the secondary cell wall. Actually, what happens means in the case of plants, for example, it is a plant cell. Uh, it has a cell membrane, and after the cell membrane, there is a primary cell wall. And primary cell wall is present in the younger plant cell actually. As the plant grow in size or as the plant matures, there is a formation of secondary cell wall towards the inner side, that is towards the membrane side, not outside. Remember this point. For example, it is the primary cell wall. The secondary cell wall is formed inside the primary cell wall, not outside. Okay, and in some cases there is also formation of tertiary cell wall, the second tertiary cell wall also, and also uh, it is a major. It can provide the plants more extra rigidity and mechanical support. Okay. Next they are talking about the middle lamella. Middle lamella actually present between the two layers of two plant cells actually. Two cell walls of plasma, two cell walls of two different plant cells. Actually, this middle lamella is made up of calcium peptides. It can act as a glue to hold the two neighboring plant cells together. The cell wall and middle lamella actually traversed by a structure known as plasmodesmatum, which is this plasma membrane. This plasmodesmata is actually similar to the gap junctions which are present in the case of animal cells. It connects the cytoplasm of two neighboring plant cells actually. Okay. Now let's discuss about the endomembrane system. Endomembrane system means endo means inside, membrane means membranous structure. Okay, it is the type of membranous structures present in the inside the cytoplasm. Okay, while each of the membranous organelles have a different function and structure. Actually, many of these organelles are function together called as endomembrane system. This endomembrane system include endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. But in the case of animal cells, this endoplasmic system is called as GERL, that is Golgi, endoplasmic reticulum, lysosomes. Because in the case of animal cells, we do not have any vacuoles, right? Remember this point, in the case of plant cells, endomembrane system consists of endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. Whereas in the case of animal cells, it is called as GERL, that is Golgi, endoplasmic reticulum, and lysosomes. Okay. Next, mitochondria, chloroplast, peroxisomes are not included in this, so they are not considered of. They are not considered of. They are not what we say. They are not considered as a part of endomembrane system. Okay, only the endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles are considered as endomembrane system, are considered as components of endomembrane system. Now let's see how this endomembrane system can work. The first part of this endomembrane system is endoplasmic reticulum. Endo means inside, plasmic means cytoplasm, reticulum means network-like structure. Electron microscopic studies reveal that there are tiny tubular structures scattered in the cytoplasm. This endoplasmic reticulum divides the cytoplasm into two compartments, that is luminal and extraluminal. Luminal means inside the endoplasmic reticulum. Extraluminal means it is outside the endoplasmic reticulum. For example, it is the luminal and it is the extraluminal. Okay, it is a structure of endoplasmic reticulum actually. The structure inside this is called as luminal, and structure outside is called as the place outside is called as extraluminal. Okay. Now some of this endoplasmic reticulum have ribosomes on it, so it appear in the rough shape. So such type of endoplasmic reticulum we call as rough endoplasmic reticulum due to the presence of ribosomes on it. These ribosomes can synthesize the proteins, and what we say those proteins can enter into the lumina of this endoplasmic reticulum. And also some of the endoplasmic reticulum do not have ribosomes on it, so they appear smooth in structure, so they are called as smooth endoplasmic reticulum. This smooth endoplasmic reticulum has a major function of lipid metabolism, sorry, lipid synthesis of lipids. Okay, and also detoxification. RER is frequently observed. RER means rough endoplasmic reticulum. They have ribosomes on it, so they will synthesize the proteins. So which cell has the majority function of protein synthesis? They have majority RER. Okay, uh, this is the point they are trying to explain here. And the smooth endoplasmic reticulum is a major site of synthesis of lipid. Just now we have discussed. In animal cell, lipid like steroid hormones, that is progesterone, testosterone, and various type of steroidal hormones also synthesized in this smooth endoplasmic reticulum. Okay. The next organelle in the endomembrane system is Golgi apparatus. This Golgi apparatus was discovered by Camillo Golgi in the year 1898. Actually, he observed densely strained reticular structures near the nucleus. So they have named Golgi based on his name, that is Camillo Golgi. Actually, this Golgi also contain many flat disc-shaped tracts. We call them as cisternae, and the diameter of this cisternae is 0.5 to 1 micrometers in diameter. Actually, these are stacked parallel to each other. Actually, the number of cisternae is varied depending upon the Golgi complex or depending upon the cell. Actually, these are concentrated near the nucleus. Actually, the forming phase, actually, it has two phases. The first one is called as cis phase. It is called as convex phase or the forming phase. And the last one is maturing phase or the concave phase. Okay. For example, if we consider the cis phase, it is called as the maturing phase or concave phase. It is a forming phase or convex phase. Okay. Or we call it as a cis phase. Okay. Uh, the cis phase and trans phase of the organelles are entirely different. Actually, these are entirely different but are interconnected. Golgi complex majorly perform the functioning of glycosylation or modifications of proteins or lipids that are synthesized in the rough endoplasmic reticulum and the smooth endoplasmic reticulum, right? Okay. So remember this point. Golgi complex major function is to do the modifications such as glycosylation for the proteins and the lipids that are synthesized in the RER and Golgi. And also, it can be helpful in the packing of materials which are synthesized in the ER. And it starts the proteins or the anything that is inside materials to be packed in the form of vesicle from the ER. They fuse with cis phase of this Golgi and move towards this maturing phase. This explains why the Golgi remains in close association with endoplasmic reticulum. Because for this reason only, the Golgi complex remains in the close association with the endoplasmic reticulum, because it needs to take the what we say vesicles from the ER and they need to modify in the Golgi complex, right? A number of proteins synthesized on the ribosomes, so it is the same point which I have told. It will be modified in the Golgi complex for glycosylation, and glycosylation is of two types actually. N-linked glycosylation, O-linked. N-linked means it involves the formation of what we say a sugar group at the asparagine residue. In the O-linked glycosylation, it involves the attachment of sugar molecules at serine or threonine residue in the proteins. Okay, so it's about the glycosylation. Remember one thing: the Golgi complex major function is to pack the materials and the modification. Okay, so it is the point they have mentioned. It is the important site for formation of glycoproteins and glycolipids. The next one is lysosomes. These lysosomes are also membrane-bound vesicular structures. These are formed by the packing in a Golgi complex. These lysosomes actually contain very different types of hydrolytic enzymes. For example, lipases, which can digest the lipids; proteases, which can digest the proteins; carbohydrates or amylases, which can digest the carbohydrates. So this is what they have mentioned. These lysosomes have the different types of hydrolytic enzymes, so that they are called as suicidal bags of cells also. Okay. Next, vacuole is a membrane-bound space found in cytoplasm. It actually contain water, sap, and excretory product and excretory products also. This vacuole is surrounded by a membrane called as chloroplast. Actually, this vacuole is occupies the 90% of the space inside the plant cells. On the vacuole, we have pumps also. They are called as V type of pumps. It will transport number of ions or the other molecules against concentration gradients into the vacuole, so that their concentration is higher in the vacuole than in the cytoplasm. And as usual, animal cells do not contain any vacuoles, but there is an exception. In amoebas, in amoeba, there is the presence of contractile vacuole. This contractile vacuole helpful in the process of osmoregulation and excretion. It is an important point. You need to remember this point. And also in protists, for example, in paramecium, we see a vacuole known as food vacuole. It will be formed by the engulfing of food materials. Next, we will discuss about the mitochondria. Mitochondria, as we all know, it is the powerhouse of the cell. We can't observe the mitochondria without staining. It is stained by the Janus green pea. So we need to stain if we want to understand, if we want to visualize the microscope, if we want to visualize it under the microscope. Actually, the number of mitochondria per cell is variable depending upon the physiological activity. If the cell is more active, if it needs more oxygen, then there is more mitochondria in it. Uh, it is sausage shape actually. Diameter is 0.2 to 1 micrometer, uh, and length is 1 to 4.1 micrometer. And mitochondria is double membrane structure. It has outer membrane and inner membrane. Inner membrane is, you see here, it is folded into structures called as, these structures are called as cristae. These cristae are present towards the matrix. Actually, this cristae will be helpful in many functions. It will be increased in the surface area, and also mitochondria has 80s type of 70s type of ribosomes. You need to remember this point. It is very important. Actually, there is a theory called as endosymbiotic theory state that mitochondria and chloroplast actually they exist as the prokaryotes, but eukaryotes have engulfed this cytoplasm into the cytoplasm, this chloroplast and this mitochondria. As a result, this mitochondria and chloroplast entered into the what we say eukaryotic cell. But don't know how far it is correct. But mitochondria and chloroplast have 70th type of ribosomes in it. Mitochondria, as we all know, it is a site of aerobic respiration, that is oxidative phosphorylation. It has this inner membrane of mitochondria, that is cristae, has electron transport chain. Just now I have told now. It plays a major key role in the oxidative phosphorylation. It can produce ATPs, hence we call as powerhouse of the cell. And also, mitochondria has its own DNA molecule and own RNA molecule, and ribosome, that is 70th type of ribosomes. And mitochondria will be divided by a process called as binary fission. It is also one of the proof that mitochondria have been existed came into the eukaryotes by an endosymbiotic theory, because bacteria also divide by binary fission. So don't know how far it would be correct or not, but there is a theory called as endosymbiotic theory. And there is one point also important we need to remember. Mitochondria in the membrane has a specialized lipid called as cardiolipin. Okay, it's all about the mitochondria. Now let's discuss about the plastids. Plastids are actually found only in the case of plant cells, and also in the euglenoids. Euglenoids belongs to this what we say protist, rhizophites, dinoflagellates, euglenoids. Uh, these euglenoids are those euglenoids. Actually, these are easily observed under the microscope because they are very large in structure. Actually, these they bear some specific pigments so that it can impart the colors to the plant. Based on the type of pigments, plastic can be different types. For example, chloroplast, chromoplast, and leucoplast. Chloroplast contain chlorophyll and different types of carotenoid pigments. It can trap the sunlight and it can perform the function of photosynthesis. In the chromoplast, actually there are fat-soluble carotenoid pigments like carotene, xanthophylls, and others also that they give yellow, orange, or red color. Leucoplasts are actually colorless plastids. They are of different shapes and sizes. Actually, they store nutrients. For example, amyloplasts store the carbohydrates. Uh, for example, starch. Uh, for example, potato. Whereas helioplast stores oils and fats. Whereas helioplast stores oils and fats, whereas aleuroplast stores protein. Okay. Amyloplast store starch, that is carbohydrates. Elioplast store oil and fats, whereas aleuroplast store proteins. Majority of the chloroplasts are in the mesophyll cells of the leaves. Just now we have initially we have seen no mesophyll cell structure. Actually, these plastids are lens-shaped or oval-shaped or discoid or even ribbon-like. They are of different shape. Okay, their length would be 5 to 10 micrometer and width would be 2 to 4 micrometers. Okay, it's all about the plastids. Next, we will going to discuss about the major type of plastid, that is chloroplast. As you all know, about the chloroplast, these are present in the mesophyll cells of the leaf. They are also lens-shaped or oval or spherical on discoid or even ribbon-like. Uh, like mitochondria and chloroplast. Chloroplast also double membrane, that is outer membrane and inner membrane. And the central space is called as stroma. This space is called as stroma, and it has thylakoids. The one single unit is called as thylakoid, and group of this thylakoid is called as grana. And these thylakoids can be connected to each other by a stromal lamellae. In these thylakoids and grana, there occurs a process called as light reaction, in which ATP and NADPH can be produced, and in the stroma there is a process called as dark reaction, in which carbon dioxide is reduced to carbon CO2 is reduced to carbohydrates, which is the basis of food for all the organisms on the earth. Okay, it's all about the chloroplast. I think I have covered all the points. It also contains the DNA, likewise the mitochondria. It has 70s type of ribosomes, and in the cytoplasm, as we all know, 80s, but only in the mitochondria and in the chloroplast, it has 70s type of ribosomes. Now let's discuss about ribosomes. Ribosomes are actually the granular structures. They are first observed under the electron microscope by George Pallid, named a scientist in the year 1953. Actually, the composition of ribosomes are RNA and proteins. They do not surrounded by any membrane. Actually, they are membraneless. Actually, the eukaryotic ribosomes are 80s. Just now we are discussing from them, and prokaryotic ribosomes are 70s. Each ribosome have larger subunit. See here, it is a ribosome actually. It contains one larger subunit and smaller subunit. When it comes to 70s ribosomes, the larger subunit is 50s and 30s. Don't add this, they are just depending upon the sedimentation coefficient. It is the indirect measure of density and size. So don't add 70s means, for example, 50 and 30, 80. Don't add that. 50s and 30s, just the sedimentation coefficient of large subunit and small subunit. For 70s type of ribosomes, large subunit is 50s and the small subunit is 30s. Whereas in the case of 80s type of ribosomes, large subunit is 60s type and small subunit is 40s type. Okay, it's all about ribosomes. Next, we will going to study about the cytoskeleton. Just like how the human being has a skeleton made up of bones and cartilages, the cell also has a skeleton structure which is known as cytoskeleton. This cytoskeleton provides has the many functions such as mechanical support, motility, maintaining of the shape. These are the functions majorly performed by the cytoskeleton. Actually, the cytoskeleton is composed of three components: microtubules, microfilaments, and intermediate filaments. Microtubules are actually very large in size. They are made up of a protein called as tubulin. Each tubulin is actually a two dimer, actually one alpha and one beta subunit. This what we say this dimer is polymerized to form one microtubule. Okay, this microtubule plays a major important role in the spindle fiber formation of spindle fibers during the formation of cell during the cell division. Okay, and also it will help in the movement of proteins in the cytoplasm of the cell. So these are the major key points of the microtubules. Microfilaments are small in size when compared to the microtubules. These microfilaments are made up of actin filaments. We'll see about actin filaments in the muscle contraction unit. It has a motor protein also, which is called as myosin. It will helpful for a long distance transport, especially in the case of plant cells. See, I have explained. See, they have explained. Motor protein associated with microfilament is called as myosin. Okay. Next, when it comes to intermediate filaments, it do not play a role in motility. Actually, it is a major important point you need to remember. Intermediate filaments do not involved in motility. They provide only mechanical strength. They are actually made up of different type of protofilaments. Okay, these are the basic points about microtubules, microfilaments, and intermediate filaments. For example, if you want to stop the cell division, there are certain drugs, for example, Taxol, uh, vinblastine, colcin. These are the drugs which will be inhibit these microtubules or microfilaments or intermediate filaments, so that we can stop the what we say cell division. So we can stop the, for example, in case of cancers, we can stop the cancers also. But if it what we say goes to another stage, so we can't stop such efficiently. So it's all about the cytoskeleton. As we have discussed initially, centrosome is present only in the case of animal cells. Centrosome, actually, I have drawn the structure. It is a centrosome, for example. In the centrosome, there is the presence of two cylindrical structures. These two cylindrical structures are called as centrioles. Okay, remember this point. Centrosome contains two cylindrical structures called as centrioles. Okay, actually, this centrosome is surrounded by amorphous pericentriolar materials. These centrioles are laid at a right angle to each other, that is perpendicular to each other. If you understand the inner structure of the centriole, it will be look somewhat like this. Actually, it has a 9 plus 0 organization where this 1, 2, 3, 4, 5, 6, 7, 8, 9, 9, right? These 9 peripheral tubules are formed of microtubules. Just now we have discussed now in the case of cytoskeleton microtubules. These peripheral tubules are formed of those microtubules only, but are three. So we call this peripheral microtubules, which are triplets. This central portion is called as hub. Hub, we call it as a hub, and these peripheral microtubules are connected to it by a stack-like structure called as radial spokes. Okay, it is a major points which you need to remember in the case of centrioles. Centrioles exhibit 9 plus 0 organization, where the 9 peripheral microtubules are triplets. So these are the points I have wrote here. It is made up of protein called as tubulin. Tubulin means microtubules. Because microtubules are also made up of a proteins called as tubulin. The central proteinaceous structure called as hub, and these are connected by radial spokes. Centrioles can form the basal body of flagella or cilia. Actually, the flagella or cilia can be arranged from the centrioles only. But flagella and cilia has a 9 plus 2 organization. These centrioles has a 9 plus 0 organization. And also spindle fibers are also formed by these microtubules, which are originating from the centrioles only. Okay. Now let's study about the cilia and flagella. Cilia and flagella are the hair-like outgrowth of the cell membrane. It is present in the eukaryotes. Cilia are small structures when compared to the flagella. These are like the ores causing the movement of the cell or the surrounding the fluid. They are present in the lungs also, trachea also, fallopian tube also. They will help in the movement actually. Flagella are comparatively longer and responsible for the cell movement. Flagella are present in the actually. The prokaryotic bacteria flagella we have discussed when we are discussing about the prokaryotic flagella. It is very much different when compared with the eukaryotic flagella. Electron microscopy study of cilia and flagella. Actually, it is covered by plasmembrane. Actually, the core is called as axoneme. It contains a lot of microtubules running parallel to the long axis. Just now I have told no, cilia and flagella has microtubules, but it has 9 plus 2 organization, where the 9 peripheral are doublets. Peripheral 9 peripheral doublets, and the central 2 are central doublets. Here there are 2 central doublets and 9 peripheral doublets. Okay, it is called as 9 plus 2 arrangement. The central tubules actually these central tubules are bridges are connected by bridges actually. The see these central microtubules having now they are also connected by bridges, and it is surrounded by a sheet called a central sheet. And also these peripheral microtubules, that is two peripheral microtubules are connected to this central microtubules by a radial spokes. Okay, it's all about the cilia and flagella. Almost all the points of cilia and flagella are covered. I have told just now, these cilia and flagella emerge from centriole-like structure called basal bodies. Actually, it is centriole only. The next one is nucleus. Actually, the nucleus is the brain of the cell. It was discovered by Robert Brown. We have discussed in shell only. Later after some time, this nucleus was stained by basic dyes because the nucleus is acidic. So it was stained by basic dyes by the Fleming, and he named it as a chromatin. Actually, the interface nucleus has a chromatin in the loose form. Actually, we will discuss what exactly mean by the chromatin in the next slide. Okay, actually the nucleus has a pore-like structure. Actually, the nucleus is double membrane actually, outer membrane and inner membrane. But at certain locations, the nuclear membrane is what we say joined. At the joint, we will observe a structure called as nuclear pore. These nuclear pore help in the movement of proteins or any RNA from the nucleus to cytoplasm, or from cytoplasm into the nucleus. Okay, and this outer membrane of nucleus is continuous with the endoplasmic reticulum, particularly RER. We have discussed all these points, uh, and also for example, some cells have more than one nucleus also, right? And also, for example, in the case of RBC, sieve tube cells, they do not have any nucleus also. It's and also in the nucleus there is a specialized structure called as nucleolus. In the nucleolus, rRNA can be synthesized. This rRNA can be very helpful in the process of protein synthesis. Friends, first let's understand one basic point. Actually, you all aware about the DNA, right? DNA is double stranded. It is a genetic material in the most of the living organisms, right? Actually, our DNA is around 2 meters long, 2 meters in length, but our cell is in micrometers in length, right? So then how we can accommodate such long DNA in micrometer cell? So for that, the answer is the DNA is coiled around a protein called as histone proteins. Around these histone proteins, the DNA will be coiled. So eventually they will be condensed and form a structure called as chromosome. It is a condensed structure of this DNA only, DNA, histone proteins and non-histone proteins, and and various different types of proteins. Okay, so it is a basic structure of chromosome. Actually, it is the chromosome which is present inside in our cells. The central region of the chromosome, or the primary construction, is called as centromere. Remember this point. On this centromere, there is structures called as kinetochores. Onto this kinetochores only microtubules can be bind during the process of cell division. Onto this kinetochores, microtubules bind and start pulling towards the poles. Okay, so actually it is a chromosome and it is a duplicated chromosome. So a lot of people get confused whether it is the chromosome or it is a chromosome. So the thing is, it is a chromosome actually. It is a single chromosome and it is a duplicated chromosome. That means during the S phase, the chromosome gets duplicated. It is a duplicated chromosome in the S phase. Okay. It was a points they have discovered. They have wrote human cell has 2 meters long DNA. we have 46 chromosomes or 23 pairs of chromosomes. These 2 meters of DNA will be accommodated in the 23 pairs of chromosomes. Okay. Now, depending upon the position of the centromere, our chromosomes can be classified into 4 different types actually. The first type of chromosomes are metacentric chromosomes. In the metacentric chromosomes, the centromere present in the middle. See here, it is a metacentric chromosome. The centromere is present in the middle, right? It will form two equal arms. See here, the upper arm and lower arm is almost same. So it is a metacentric chromosome. And also the second one is submetacentric chromosome. In this type of submetacentric chromosome, the centromere is located slightly away from the middle. See, it is not exactly in the middle, it is slightly away from the middle. So it can form one shorter arm and one longer arm, right? So next in the case of accucentric, centromere situated close to one end. So see here, centromere came to close one end, close of one end, right? So here the formation of one extremely short arm. So it is very extremely short and very long arm. See here, one very long arm, right? So in the telomeric chromosome, the centromere is located at the terminal position. That means at the last position. It can form at least a small short arm and the very long arm. See here. And also a few chromosomes. For example, see here, this is metacentric chromosome. It has this non-staining secondary construction also. Primary construction is centromere, right? And it has a non-staining secondary construction also. It is called as satellite. Okay, this giving appearance of a fragment called appearance of small fragment, and it is called as satellite. Okay, remember these points. This may ask in the exam also as a question. And the last thing is microbodies. Microbodies are the membrane-bound minute vesicles, and also they contain various enzymes also. These all formed from the Golgi only. These are present on in the plant cells also, animal cells also. So it's all about the cell the unit of life. Complete chapter of class 11 NCERT biology. I hope you got understand. If you understand, share your feedback in the comment section. Thank you.